Cell differentiation is the process by which a stem cell becomes a specialized cell type. This happens naturally in a developing embryo. This is an embryonic stem cell. It is pluripotent, so it has the potential to make all cell types of the body. As it differentiates, it loses this potential and becomes more specialized. A cell's location within the embryo and chemical signals will determine which of the three layers, known as germ layers, the cell will become part of. These three layers are the endoderm, mesoderm, and ectoderm. Once a stem cell has differentiated into a germ layer, it is now multipotent, which means that it can make many cell types within the layer. The cell experiences many genetic and physical changes along the way. As it differentiates, the cell becomes more specialized, and the number of cell types it can turn into gets fewer and fewer. The cell is gradually losing potency. Now this cell is unipotent. It can only become one cell type. Within each layer, a cell can differentiate into a number of cell types which are unique to that layer. The outermost germ layer is called the ectoderm. An ectoderm cell can become many different cell types, from a skin cell to a neuron. The middle germ layer is called the mesoderm. A mesoderm cell can become all types of muscle cells, such as heart or skeletal muscle or red blood cells, among others. The innermost germ layer is called the endoderm. An endoderm cell can become many of the cells in our organs, such as the lungs, thyroid, or pancreas. Differentiating cells in the lab. Researchers can replicate this process in a lab. They start by putting stem cells in a culture dish. Then they add growth factors, which are naturally occurring proteins or chemical molecules that change the activity of the cells and cause them to replicate into a different cell type. They repeat this process each time with a particular combination of growth factors. At every step, the cells are becoming more and more specialized. Finally, the researchers have made skin cells. Inducing pluripotency in the lab. By adding a combination of 4 proteins known as transcription factors, scientists are able to convert normal adult skin cells into pluripotent stem cells. The transcription factors remove the skin cells' identity markers, resetting them to a pluripotent state. These kinds of engineered stem cells are known as induced pluripotent stem cells, or iPS cells. And, like other pluripotent stem cells, they have the ability to make all the cell types of the body. This is like pushing development back in time. This outstanding scientific discovery earned Shinya Yamanaka a Nobel Prize in 2012. This revolutionary advancement is leading to research and treatments for many diseases.